time is not just a measure of motion. For philosophers like Aristotle, time was more than just a rhythm on which the celestial stardust danced beautifully. Much before Greek philosophers, the rishis of Bharatvarsh praised the superconsciousness as Kalatrayatitaha, meaning beyond time. Today, the life science has approached a state where we can measure the leaps of a paramecium under microscope due to the movements of cilia. This is a different cosmos, the microcosm. Just like the vast spread of cosmos, but this microcosmos, the microcosm, is invisible to unaided eyes. And yes, the microscopes help to explore this microcosm. Hello friends, this is Lakshmi Raman Oza from Samarth Biology Tutorials Latur, and you are watching SBTL webcasts. In comparison to the diversity of the life, the unity of life is remarkable. All life forms on this planet are made up of fundamental structural and functional units known as cells. In fact, we all began our lives as a single cell. A cell that was formed in the fallopian tube by union of two pronuclei, male pronucleus and female pronucleus. Eventually, we transformed into a complex form through a process of gestation in a period known as gestation period. All of us, including the other forms than humans, they are all made up of 4 nucleobases. Those make the blueprint of our structures, our functions. For example, all living forms, including bacteria, have a large molecule known as DNA molecule. And every DNA molecule in every living form on this planet is made of just 4 nucleobases: adenine, guanine, cytosine, and thymine. All we livings on this planet, from a bacterium to a multicellular form, a giant form like elephant, all these life forms on this planet, they receive the energy required for biological processes by a common process. That is what we call as glycolysis, breakdown of sugar into 2 molecules of pyruvic acid. Look, from bacteria to an elephant or a whale, this process is common. Even in the plants, this process is common. At large, we share common characters more with all life forms than the variations we possess. And this becomes more and more visible as we enter the microscopic world. For example, the process of expression of genes that takes place through two processes, transcription and translation, in all living organisms. Bacteria also are no exception. The process of expression of genes, what we call as protein synthesis, it needs some structures in the cell known as ribosomes. So these ribosomes, which are protein factories of cells, they are common. In all living organisms, the proteins are made using these ribosomes. Look at this remarkable unity in diversity in all life forms on this planet. But when we are questioning, when we are trying to solve this riddle, what is life? Then let us understand one more example in biology, and that is virus. Viruses have a proteinaceous covering known as capsid. These capsids are made up of some units known as capsomeres. And other thing than this capsid is viruses have their own genome that may be either DNA or RNA molecule. But viruses lack ribosomes. They do not have their own protein synthesis machinery. So here the viruses are pushed outside the living world. They are said to be at borderline of living and non-living world. We don't say virus cell, but we say virus particle, because viruses are not alive outside their host. When viruses infect the host, they become alive. Means what? They use ribosomes of the host to synthesize their own proteins. Now they are alive within a cell, but independently they are not alive. So essentially at molecular level, if we think, this presence of ribosomes is an important thing. This is a prerequisite for making a cell. So life forms, they share lot many similar characters. As an example, bacteria have ribosomes called 70S ribosomes, which we will discuss later on. And eukaryotes have 80S ribosomes. This S represents Swedberg unit. You see, in most of the physical quantities, the names of scientists are given. For example, when we say force, its MKS unit is Newton, name of a scientist. There are lot many names of scientists. Those are used to define a unit in physics. May that be Ohm, may that be ampere, may that be even volt or any kind of these units. Now in biology, this unit, Swedberg unit, that is also named after a scientist Theodor Swedberg. And we are going to talk about him in some forthcoming parts of this series. So, living organisms share the common characters more in comparison to the variations they have. They share a remarkable similarity. They share a remarkable unity in comparison to diversity. And that we are going to explore, and we are going to understand what exactly does it mean by immortal cells. Are there some cells which do not die? Are there some animals which regain their original form along with the succession of their age? Do they return to the original form? We are going to talk about some examples like this also. So, let us enter the subject now. What one could see in ancient past: the sky, the soil, the rivers, the horizons, lions, and even an ant. But hidden in this magnificent fabric of living world, there is a cosmos, a microcosmos, a microcosm, invisible to eyesight. Life science took a sudden twist beyond eyesight with a new vision in 17th century. A Dutch draper, Antony van Leeuwenhoek, observed microbes first. Swarms of little eel-like microscopic structures, which today we call as bacteria. But it was not till 19th century that science began to piece together the puzzle of cells. It was based upon many independent microscopic observations. Schleiden, Schwann, and Virchow, visionaries in their own right, they stitched a truth: the cell biology. Livings are made of cells, from a microbe to giants. Today, our lenses are powerful. We map genomes, we witness organelles traveling on the tracks of microtubules within a cell. None of these would have been possible without a unique human trait: a vision beyond eyesight. Helen Keller once said, "The only thing worse than being blind is having eyesight but no vision." And in Journey of Microscopy, it is this vision, not the instrument, that has unlocked the wonders of microbial life. Not a philosopher, not a medical person, but he was a cloth merchant. He used a lens to observe the knitting of fibers in the fabric. A.V. Leeuwenhoek, a Dutch draper. Here begins the first phase of cell biology. He observed many things under his simple microscope. In those things, there were some raindrops, there were some mud extracts. Even he saw the spermatozoa of a patient with gonorrhea, samples collected from a patient with gonorrhea. Here begins the first phase of cell biology. Leeuwenhoek named those microscopic structures as animalcules. He thought that perhaps these animalcules, those are seen under microscope, these animalcules, the suffix cule, that means small in size. For example, in science, we use mole as a unit of quantity, and from that the word molecule is derived. In similar way, animalcules are microscopic animals. That time, the concept of fertilization was not clear, that male gamete and female gamete come together. Instead, the idea of animalcule that was given by Leeuwenhoek, that was something like that. The leavings are preformed as a structure and they are in the form of animalcules. These animalcules, they just grow in size inside the uterus and they can transform into a complete living form. But later on, when fertilization process was described, this idea was removed from science. Second thing, Leeuwenhoek sent all his observations, their details, to Royal Society in London. This was a community of scientists in which there were scientists like Isaac Newton, Robert Hooke, and many more. His observations were not worth ignoring, although he was not a man of science. He was simply a cloth merchant. And that is why his opinions were not rather ignored, but his scientific observations and the sketches he had sent to them, those became a matter of curiosity for scientists. Those were part of this Royal Society of London. And in that, there was a group that was Academia de Lensae, a group of scientists who used to study the lenses, the properties of lenses, optics. Robert Hooke was one of them. He also constructed a microscope under which he saw a cross-section of cork, and he first of all used the word cell in his book Micrographia, published in 1665. So these two people were pioneers of cell biology, Leeuwenhoek and Robert Hooke. In 1772, a physician from London, his name was Benjamin Martin. He speculated a possibility of diseases caused due to microorganisms. Those could be seen under microscope. But it was not till the year 1864 when Robert Koch and Louis Pasteur, they gave the germ theory of diseases. This second phase of cell biology begins with the work of three great people, M.J. Schleiden, Theodor Schwann, and Rudolf Virchow. It was thought that cells are produced from nucleus, which was described by Robert Brown in 1831. So Schwann and Schleiden, in their cell theory, they borrowed the same idea. Cells are produced from nucleus that was described by Robert Brown, and that is why it was named as cytoblast. Blast means a structure which produces some structure ahead or further. This misconception in science was something interesting that is known as spontaneous generation theory. It was thought that these microorganisms, those are visible under microscope, they are produced from non-living material, and those are produced through a spontaneous generation. And this spontaneous generation theory was disproved through the observation of Rudolf Virchow. He said that cells divide and they produce new cells. The idea of cell division came through the work of Rudolf Virchow. And this is the second phase of cell biology. Now we begin with the third phase of cell biology. This third phase of cell biology begins with an extraordinary idea, that is germ theory of diseases. Robert Koch, father of bacteriology, in 1864, he suggested this idea of germ theory of diseases. He said that we must be able to extract a microorganism from a diseased person. That sample we must be able to independently grow in the laboratory. If that is injected further into a healthy individual, then symptoms of same disease, if they are expressed there, then that microbe is causative of that disease. Now an interesting thing comes here. You are collecting a microbe. You are culturing that into a lab. So here begins the third phase of cell biology, and that is cell cultures, tissue cultures, etc. When you take a cell outside the living body and use nutrients to keep it alive, then for how much period it can remain alive? For how much period it can continue to grow? Does it grow continuously without any limit? So the cell culture experiments began in the third phase of cell biology. And this was an interesting thing. Now, if we go to the fourth phase of this cell biology, then this is a molecular biology phase. Cells where the minute parts of cells at nanoscale one could observe because of invention of that electron microscopy. Here, science took a new turn, cell biology took a new turn. And scientists even noted that cells have some specific kind of behavior. They move towards a nutrition, they move towards a nutrient particle, and they move away from a toxic substance. The Nobel Prize in 1908 was given to Mechnikov, and he discovered something called phagocytosis. The cells in our body, they detect a bacterium, a harmful toxic substance, and they eat that substance. This process is called phagocytosis. So the study of cell became more and more comprehensive eventually. It became more and more useful. Then the studies of cancer, they became more and more comprehensive. Success of a cell depends upon two things. The first one is how it gets its nutrition. And the second thing, how the cells maintain a good lifespan. Cells do have their fixed lifespan. For example, a red blood cell, it survives for 90 to 120 days in our body. Many different cells have different lifespan, and after that they die. This natural death of cells is known as apoptosis, programmed cell death. Sometimes this death is also due to some injury or some toxic substance. This death is known as necrosis. In fact, the life of a cell in a healthy manner is important. It should remain alive for a longer period. It should not die early. But sometimes an injury or some toxic substance or infection that would result into death of cell. What if a cell is immortal? The cell does not die. Yes, it is found in a disease called cancer. The cells do not undergo apoptosis, a natural death. They continue to live and divide, and they form a lump or a tumor. In fact, longevity of a cell is an important factor, but in case of cancer, this becomes dangerous. The second factor for successful survival of cell is the cell should get its nutrition from any blood vessel that passes nearby it, a capillary that surrounds it. The newly formed, starved cells in our body, they send molecular signals to nearby capillaries. These capillaries extend branches towards the starved cell, and this process is called angiogenesis. Unfortunately, in case of cancer, there is angiogenesis that results into formation of new capillaries surrounding the cancerous cells. That ultimately results into spread of cancer in the body. In fact, the cells are immortal in case of cancer, but this immortality is cursed, just like the character in Mahabharata. His name was Ashwatthama, an immortal being, but a cursed immortal. Utpatti, Sthiti and Lay, birth, being and death. These are used for describing the life in Indian scriptures. Thank you for listening me. This is Lakshmi Raman Ozha and you were watching SBTL Webcasts.